but for the most part, I think the rules are better followed by machines. And so now we have probabilistic genotyping, which many times I know your M-Vac is used to collect data, and it's sometimes very complex data. It seems that the probabilistic genotyping is better than, or certainly helps people, helps me when I'm doing manual interpretation. Now, I'm also finding some problems with probabilistic genotyping. No matter what we invent, whatever drug you invent, there's a side effect.
And, yeah. Well, before we move on,
yeah.
For somebody that doesn't understand, like if our audience was a jury, for example,
yeah,
explain probabilistic genotyping.
Sure. So when we get a mixture of DNA that's more than one person, and I think with with what your product, the M-Vac, you often get that because you'll get deep residual DNA from fabric or something, from a couch. And when we have that, it looks like a lot of peaks on a graph. And what we're trying to do is to group the peaks into single people, into representative people. So all the peaks that are about the same height are possibly from the same person. If there's a lot of tall peaks on the graph and a lot of short peaks, maybe it's two people. And so it's the amounts of DNA that we see at testing many sites. What probabilistic genotyping does is it actually generates kind of random interpretations of what that can mean and tests them. Is this random interpretation better than the one I just did? And for me to do that by paper, it's an hour and a half, 2 hours for each of those interpretations. Whereas the computer can do, 100,000 of them in an afternoon, right? And that's what it is. So it's calculations, comparisons of hypotheses, basically, many, many, many hypotheses to get to the most likely, the interpretation that says the evidence is most likely with there are two people in this mixture. Greg Hampikian, put in the most, Jared put in the lower amount. That's the interpretation that works best. We tried, Richard Nixon, we tried Obama, Trump, none of them are included. Right. And that's what the computers are good at, is entertaining lots and lots of hypotheses and then telling us which ones, for which ones is the evidence most likely.
Right. And that's the interesting thing about that, especially in conjunction with the M-Vac, because the M-Vac collects so much more than other collection methods. Like you said, oftentimes it can get deeper, it can go wider. And so you may get more DNA, and some of that DNA is your target DNA, but some of that DNA might be DNA you don't necessarily want. But fortunately, because of the advances in the lab processes, and again, because of this probabilistic genotyping, the software that can interpret it, it enables us to become more clear on possibly what those results were. And so, but the interesting thing is, even with that, you still have to take those results, give it back to an investigator and say, now, here's some more actionable data. Now move forward with it and continue on with your investigation.
Right. And then eventually you have maybe a Daubert hearing, which, there's two ways that we consider evidence. One is, will the evidence be presented to the jury or excluded? And that happens without the jurors there. That's a hearing that can happen. So a judge has to be convinced that the level of the science is generally acceptable, that an error rate has been established, that it's been peer reviewed.